Thank you for inviting me to this fascinating conference and for the opportunity to introduce our panel discussion. I left Nature Genetics after 15 years, encouraged by the fair data movement that it would be possible to get more and better papers if we pay attention to metadata and explicit models around the metadata. So that's the hope, that I will be able to attract perspectives and analysis papers from you and your collaborators to my journal at Wiley, Advanced Genetics. What I'd like to talk about today is a class of digital objects that I think will make a big difference to data sharing and to the generation of semantically correct and more useful papers. What do we know about ATP? Well, it's clear that the number of publications about ATP increase enormously, and that there's no way that one person can get their head around all of this information. So the idea of a digital fair molecule is to build an avatar of ATP that can find all the information about the things that ATP is and the things that ATP can do, from energy transformation to protein phosphorylation to nucleotide synthesis. I've been working with the FAIR Data Group led by Eric Schultes and colleagues. The actual infrastructure for digital objects is being done elsewhere, but the FAIR Data Group is particularly interested in building digital representations of molecules. So here is an example of a paint chemical, a ester of linoleic acid found in fine paintings, represented in ones and zeros. There is extensible metadata which tells you its provenance and what it's used for and where it's found, and of course the unique identifier. We've written a preprint about this, and there's some examples. A smaller example would be the problem of chirality. This is a familiar amino acid, threonine, and it comes in 4 isomers with different chirality. And by representing the chirality with ones and zeros, along with the digital representation of threonines, carbon backbone, side groups, and all the rest, we can, with the digital object, correct the confusion that occurs in various chemical databases as to the chiral centers that are involved in all 4 of these isomers. So you can see here the representation that finds that there's often wrong representation of these isomers. So that's just a simple example of what you can do with a small digital object that is a fair molecule. I'm much more interested in the use of fair molecules in genetics. For example, this fair molecule represents any microRNA, and there's huge confusion about naming, roles, methods, and publications, including quite a lot of fake papers in the field of microRNA biology. And I think that this semantic naming would allow the digital objects to help make some sense of this literature and of the databases that represent the findings of this field. Familiar to everybody and present in almost every genetics paper are a pair of primers, which are the brackets for extracting by PCR any piece of DNA from any genome or reverse from RNA for qPCR. So the amplification primers that identify a unique piece. And of course, you can use a similar approach to generate a generic splice site in a molecule of interest, like the breast cancer gene BRCA1, and then use that to represent all the possible mutations that influence the splicing of that particular exon. Structural variants, transposons, chimeric reads, artifacts, cloned sequences, fusion constructs. This is a generic molecule that identifies a lot of molecular biology products. This is a pair of SNPs that are allelic to each other. The SSID has been abandoned in favor of the canonical RSID, but the SSID is actually a slide rule that uniquely identifies the position and identity of a single nucleotide variant. For GWAS literature, genetic epidemiology, mutation research, this is invaluable. This is a slippage repeat that causes the neurological condition Huntington disease. This is a slide rule that will find that in any bioinformatic project, a machine can now identify the length of a pathogenic or harmless triplet repeat using this construct. Position of modified bases for methylation, epigenetics, anything you want. Again, these are generic fair molecule templates. I'm interested in using fair molecules to go across data sets to carry out experiments in genetics that are bigger than can be achieved in a single lab. And relevant to this day's conference, RNA and peptide vaccines, glycosylated antigen vaccines for SARS-CoV-2. This is a paper from Cell that talks about the antigenicity of the spike glycoprotein, and here are some other papers that could be integrated using a spike glycoprotein or spike molecule and its target, ACE2, in the host cell. So neutralizing antibodies here blocking the virus entry to the cell, the single cell sequencing of B cells that produce those antibodies in people who've recovered from COVID, and a hamster model to test immunization against this particular antigen for clinical trial proof of principle. Here are some other examples that interest me, but not relevant to today's conference. Activating T cells through a receptor using non-peptide, but ribonucleotide small molecules, rare variants in breast cancer. I mentioned the splicing example. I mentioned the Huntington's disease example. I think all of these would enable an entire community to work on the same objects and to build knowledge in a much more interoperable way than we currently do. Again, relevant to today, I think another paper from Cell illustrates where semantic precision can enable collaboration. And this is where variants in the SARS spike protein interact differently with host protein variants in the receptor and the processing molecule protease TMPRSS2. And the available data on variation can be found here for ACE2 for humans, and here for TMPRSS2, again, for human population variation. You can also look at the variation between species, the fixed residues that differ between different mammals. That's relevant to host range, whether the virus can infect particular hosts like ferrets that are particularly susceptible, or other hosts that are relatively resistant like bats. It's possible to look at the standing missense variants present in the human population. Here they're evaluated for whether they damage the ACE2 protein or not. And underneath are the allele frequencies in various human populations. It's all very well to have a semantically correct and data-based set of data representing human genetic variation, but I think quality measures are key to its reuse. Here is an example of two variants from the NOMAD database, which is superb because it shows quality measures as well as the relationship of this database to others. And what you can see is this is a coding missense variant that's probably damaging to the protein's function. The genotyping quality is exquisite. The quality metric is right in the middle of the highest quality range. I think this could be used in a study because there's a lot of available data and it's high quality. Here is another variant that's very interesting because obviously it varies from East Asia, where the case fatality rate is very low, to Europe, where the case fatality rates are very high, and the variant tracks very closely to case fatality rate. So you'd like to do an association study on this variant. However, there is a warning in NOMAD that the genotyping quality is very poor and the site call rate is excruciatingly low. Fewer than 50% of individuals have this variant covered. And so although it's exciting looking and its distribution is interesting, the quality of the data supporting any use of this particular variant comes with a health warning. All right, that's all I'm going to say about COVID, But now there are some other examples where I think fair digital molecules, digital object molecules would be extremely useful. RNA modifications, there are enormous numbers of these. They live in a very large number of databases, and most of these databases are either inaccessible, interoperably impaired, they don't talk to each other, and the number of such databases involving nucleic acid research is, of course, increasing at a horrific rate. So I'm hoping that some buy-in and some digital object infrastructure will enable a whole slew of very high quality analysis papers to be generated using digital object fair molecules. And if you have any such ideas for papers, either perspectives or analyses or research articles, get in touch with me and please submit to my journal. We're very quick, we're very experienced, and we would love to see your work. Thank you all very much for the opportunity, and I open the panel.